I think despite the third wave of the corona and the Omicron, I think many people have gathered here to listen to how to write a thesis paper. Even though I think I can see only 6-7 people are there, but still I think it's heartening that at least some people are interested in writing a paper. Most important thing is everything. The thing starts with a thesis commonly. I thought India possibly will be doing a large number of MD and DM level thesis, which actually goes to the junk. It absolutely goes to the junk, and hardly less than 1% gets published. Because the problem is that at the thesis stage, it is not properly addressed. All the issues, various lacunae are not thought for, and time is not given. Actually, adequate time is not given in writing a thesis. And then whatever results come, you can't control the results. Like, the problem starts in writing the first draft of your thesis protocol. That is where the problem comes. If you want a good paper, we need to improve how to write a thesis protocol. So I will be focusing on this, and then a brief mention about how to convert the thesis into a research paper. Because writing is an art. The writing, I think we can talk about writing, scientific writing for many classes. But for you people, I think the most important thing is how do you put together a good thesis protocol. That is the most important thing to start with whenever you want to do a research. For that, this is a very good starting point. And then most of you have done a thesis at the MD level also. And people who have done it from a good institutions, they always know the various methodology that are involved. There's rigorous scrutiny that thesis presentations, thesis protocol presentation that happen, and various faculty they contribute, and you know the basic background. So this is for those people who are not exposed to such rigorous thesis protocol presentations. Many of you are in DNB situations where I think such rigor is not there in developing your thesis. So again, most important thing is choose your topic and writing your thesis statement. So what is the thesis all about, and creating an outline for the not only the protocol but also for your final paper. So this will be the starting point. Like, choosing the topic, that is I think the most important thing I will be mentioning about. I think you have to be realistic. You can't solve some of the problems that are not being solved by the great academicians of this country with the thesis, most often than not. But there is no harm in starting an effort. I told you, sir, you do a bad job or do a great job, the amount of time that in developing a good protocol, in doing a thesis, is going to be the same. Writing a crab 100 pages is the time taken for a crab is more than if you do a good job. You like that good job, and then you spend more time and you get a good thing out of it. So doing a good thesis, a good protocol is more productive and possibly yields more for the amount of time you spend than a bad protocol. And once you have written a bad protocol, you started with a bad thesis, a bad question, then possibly nobody, even the best writers in this country or in this world, cannot revive this paper. The paper is dumped. It is worth for only junk. So most important thing, the common mistake that oftentimes happens is that the enthusiasm of youth, and they want to answer everything. Like, so for example, one of the thesis done by one of my senior resident was a prospective observational study of aortoarteritis. So this was a very good study. Like, there are not much data available in children. Okay, so in children, prospective study of is very important. So, but what in prospective study? What in prospective study? Can anybody say something like, because it's a Zoom meeting, we can interact. So what in prospective study of aortoarteritis in children? Because at AIIMS, we get a lot of aortoarteritis in children. They often present with acute decompensated heart failure, severe LV dysfunction, and they have sudden occlusion of or acute or subacute involvement of the abdominal aorta, as well as the thoracic aorta, as well as the renal arteries. So we have a huge collection of cases. So we wanted to do it prospectively. So what will you focus for that particular thesis? What will be your focus? Anybody wants to answer? What should be a good focus? So it's a prospective study. You can talk about the way how the patients are presenting. That can be a prospective study, or how the patients are responding to your initial treatment. How many of them are active? How many of them satisfy the criteria for aortic arteritis? So basically, it can focus on diagnosis. It can focus on the yield of, for example, the yield of routine screening of all early dysfunction with a Doppler or an echocardiography and Doppler. So you can do many things in prospective study, but the most important thing will be what are the short-term and medium-term outcomes. Once, for example, rather than making, I just got management outcomes. Yeah, that's right. So like that Harsh has written that it's management outcome. So you can put it in chat box or you can discuss. So like the focus was the short-term and medium-term outcome of children presenting with aortic arteritis, a prospective observational study. So look at a prospective study of aortic arteritis. Now we have got a more clarity. Like the more you think, the more clarity you get. So what we need in the starting pages, the starting first few sessions that we'll spend will be to get the clarity in research. Once you are clear about what you want from that particular study, everything falls in place. So that turned out to be a very good study, which I think got published in BMJ Pediatrics. So that's how we go. So a simple thesis gets published into a BMJ group of journal with a high impact factor. And for example, this is not about boosting. I'm not here. I have published enough, and I'm not here to boost. But I'm trying to like ignite a fire in among the young pediatric cardiologists of this country that we need to solve our problems. Nobody else is going to do a research in children with rheumatic heart disease, children with children with aortic arteritis, and late presenters with various diseases, Eisenmenger syndrome. We are the capital. We are the capital of Eisenmenger syndrome. Why can't we do a good thesis on Eisenmenger syndrome that will get published in a good journal and you answer a good question, rather than focusing on something that has already been done? Like oftentimes I find a lot of theses come to me for evaluation of the DNB board. So most of the time what we do is already a study has been published. It is well published, and maybe 2-3 studies are there. First study gets published in a very good journal. Second study gets published in a regional or local journal. Then now you're actually replicating the same work. So we are doing what is known as research. We are searching, because only thing is it has never been done in India. So we want to do this. So don't do this. The amount of time you are going to spend on such a topic and is going to be nearly the same. And the wasted time is much more if you do a bad study compared to a good study. And we'll be going to the thesis statement and creating an outline in the subsequent slides. So for example, I was telling about like last few years have been really productive for us. Some of the theses of ours have got published got various awards, and has got published into good quality international journals. Like for example, the JAG group of journals, I think 2 people have published their thesis in the last 2 years I'm telling about in pediatric cardiology, and then one in the heart. The BMJ heart also it got published. So I think it is not impossible for us to get a good quality work as a part of your thesis. My thing is that always thesis is done, boss. We need to get thesis done and finish it off because it's a regulatory requirement. Don't do this. Like DNB in a super specialty is almost equivalent to a PhD. So you're supposed to be an authority in at least one field, and that field should be wherever you have done your thesis. So I told you, like, choosing the topic is very important. And I always feel that you should not do, because publication is always a goal, but publication should not be the only goal. So it gives a lot of satisfaction. Why you do research? Majority of Indians do research. Indian researchers do research because it is all self-motivated, and very few do research because they need it for promotion. So that people who do for promotion, they finish at 5 or 10, whatever be their deadline number that is there. They don't go beyond that. So do it for it's a passion. So develop the passion at a younger age. That's a very important thing. So all of you are in that level where you should develop a passion for research and a passion to solve our own problems. That is the most important thing. Once you have that as your basic goal, everything else falls in place. You always look for a field that is of importance, that is of relevance to this country, to this local place, wherever you are working, to the people, the public that you're dealing with, and to the population, the children that you're dealing with. It has to be relevant and it has to be important. Always think of an original idea. So there is, I told you, there is no point in doing a research. Always think of an original idea. And sources and resources, that's very important. You can actually think of something great idea, but you should be able to execute it to perfection. Ultimately, the bottom line lies in execution of whatever ideas you have. So there, I think the guide plays a major role, what is possible and what is not possible within the available sources and resources that you have. So oftentimes you can have a great number of patients you'll be having, but you may not have the particular expertise in the particular thing that you are going to have. So depending on everything, what is the importance of this field and the question that you are asking, what is the relevance to the country, the population, and think of an original idea and get it focused. Get into a particular focus and then see that whether you are able to handle the sources and resources. And nowadays it is very simple and very easy. Now we have Zoom, so you can call for help. You can have a resource person. For example, like I've been part of some other thesis which are very good, which got published in a good journal where we used 3D echocardiographic strain and other things, where the particular person who had the machine but who did not have the expertise. So he came to me and then we taught him 1 or 2 times, and then later on we interacted by electronic medium even before the Zoom era came. So I think we were able to get a good workout of it. Like. So it's about that passion and the desire to get an answer. It's a particular desire to get an answer. So what do you want to focus on? So focus on 1, the population, the place, and the time. So you particularly define, for example, you want to study the outcome of device closure of VSD. This could be one topic, like, because VSD closure is not, if you are a large volume center where do a lot of VSD device closures, this could be a good topic. But then let us get the focus right. Let us get it more defined. So one is whether we are going to study them prospectively or retrospectively. So a prospective study of the word comes. If you are going to prospectively study in whichever institution of perimembranous VSD undergoing a device closure, or you want to critically say in children or beyond infancy, where you want to define over a 2-year period. So think of it like your thesis title was study of outcome of perimembranous VSD device closure. That could be one topic. But think of it, the title is a prospective study of short-term and medium-term outcome of children undergoing perimembranous VSD device closure over a 3-year period. So this actually, in nutshell, you thought about every possible thing. You're not going to study about muscular VSDs, which I think where possibly device closure is more well established. You're not going to include adults with congenital heart disease, adults with VSDs who are going device closures. You're not going to, it's a single center prospective study. So get the things into focus, get it more defined, well defined. So in today's time, many of your friends are there, even your thesis guides friends will be there. You can actually now think of combining even during a thesis time. Or you can do 2 centers can come together and contrast their results. Those things can be part of 2 theses like. So these things will add value to the thesis that you are doing. So always think of these lines also. If the population is not enough. So how do you know the population is enough or not enough? So what is the outcome? Suppose, for example, we take this medium term results of VSD closure. So you are there for 3 years. You can do, suppose your center does some 50 odd VSD closures in a year. So you will have 100 patients in 2 years, which you can follow for 6 months and produce a very good thesis. 100 prospective children undergoing VSD device closure, and what are the echocardiographic outcomes? How many develop AMR? How many develop AR? How many develop residual flows? How many have all those things you can note down? So how do you know whether that population is enough or not for a good scientific study? How do you calculate a sample size for such a thing? So you need to identify, for example, if you are, for example, in this thesis, your job is to identify the complication rate of medium-term complication rate of VSD device closure. If you expect the complication rate to be 2%, there is a way to calculate based on the prevalence what should be the sample size. If that sample size is not adequate, possibly you should do a combination. If it is really not adequate, then possibly there is no point in doing the study. This is how you should get your focus right and get your things going. So all this I'm saying before you start your thesis. Oftentimes we have 3 or 4 options for thesis and we don't know how to choose them. How do you choose? All the common questions can be easily asked to your topic. Which one to do? Based on what are your answers to why should I do it? Because it is your thesis. Yeah, your thesis guide can give you some 4 options or 5 options, or maybe give you two things that this studies have been happening. You just continue the study. That I think is you should not accept, because this is your thesis. Wherever you go, suppose if you are ambitious, you want to go abroad, this is your thesis. This is your work. It will always remain your work, first research. So always think of something that is passionate. Why should you do it? You can think of an award, an international conference presentation, all those things. But most importantly, try to answer a pertinent question. That is very important. Always the question comes, whether should I do this work or not? Always think of it 3 years down the line, once you're finishing it off, 2 years down the line, whether that will get accepted in any award sessions, even in PCSI. Think of it. You know what kind of award papers are presented in PCSI. Just think of it. If it is not, please don't do this. Don't waste your time in this. So that's a very important thing. I think that you should go on. So what are the basic components? Basic components of a thesis protocol is introduction, the review of literature, aims and objectives, and a broad thesis statement that you are going to write. So introduction, obviously, why are you going to do this? What are the importance? In nutshell, what is already there existing, and how this study is likely to add. What are the lacunae in the literature, and how this study is likely to add, and what is the importance of this? So this has to be in brief. You have to mention this. And then most important part is the review of literature, as I think which we have discussed last time. So how an extensive review of literature is a very important starting point. And always remember, whenever you are doing an observational study or mainly a randomized control trial or an outcome-based study, always define your primary outcomes and secondary outcomes. So for example, we published a VSD, propranolol for heart failure. So what could be the outcomes in this propranolol we have given in all children with VSD in a randomized fashion compared to conventional therapy. So propranolol was given and other group did not receive propranolol. So the endpoint that we can consider are: 1, admission for heart failure, weight gain, death, pneumonia. So many things can be kept. And oftentimes, the earlier way of research was you collected all the data and later on you just mix match and whichever is suitable you used to present. This used to happen maybe some 20-30 years back. But now you cannot do this. Now you have to be clearly defined primary endpoint. So in this particular study, we kept a primary endpoint of need for surgery, death and hospitalization for heart failure or pneumonia, a composite endpoint. That is the primary endpoint that is we have defined. So what is the importance of a primary endpoint? Such a defined primary endpoint is based on that defined primary endpoint, you are going to calculate the sample size. So for example, in this group, how many are going to end up having this composite endpoint? And in the other group, how many are going to get the composite endpoint? Based on which the whole study design is calculated. Whole study is planned. And can you change it later on? You cannot change it. How will other people come to know? Because nobody accepts unless you register your study in the various portals that are there. For example, in a good observational study, even it needs to be registered, especially multicentric studies. All the randomized control studies are supposed to be registered before the first patient is enrolled. The whole protocol is put either clinicaltrial.gov, the US websites or various such websites, or we have the ICMR site where we have to put this is the study, this is the population, this is our primary endpoint, and this is how the sample size is calculated. We are going to do so many numbers of patients. Without this, no journal is going to accept it after 2 years once the study is published. It's going to be ready and getting published. So that is the important thing. So like the thesis statement should have a defined population. You should have a population, the place, the time duration, what are you going to study? Everything has to be well defined. So these are the various things that has to be thought out when we are planning for the materials and methods. What is the exact population that we are going to study? What is the type of study? Whether it is an observational study, whether it's a randomized control trial, whether it is a cohort study, whether it's all those things are there. There are various types of study, whether it is prospective or there is a retrospective study, and what is the time period of study, and what are the defined outcomes. Like, for example, it's so simple. It looks so simple. Like, for example, in the BSD PHF study that I was discussing, we said this heart failure hospitalization. Are there any definitions available for children with advanced acute decompensated heart failure? The definitions are not there. So you have to define it carefully, because these patients are seen by multiple physicians. It has to be same across the field, and those definitions have to be already entered. So in the material method section is the most important thing that possibly will define whether the paper is going to be accepted or rejected in the later. This is we are talking about thesis protocol writing phase, whereas when your thesis gets rejected, this is when possibly it will get rejected. So like if you are using molecular methods, the detailed molecular methods has to be described in the thesis protocol itself. Same way, if you are doing an echo-based study or an MRA-based study or whatever investigation-based study, what are the exact echo machine that is going to be used? How the patient is going to be sedated? What are the parameters that you're going to test? What are the views you are going to acquire? How are they going to acquire it? Who is going to acquire it? How are they going to be analyzed? Everything. This is the most important thing that you have to think over and put weightage. And oftentimes when you're doing something that is unconventional in echocardiography and all, like for example, more than ejection fraction or volumes and dimensions, it's always a better idea to plan an inter and inter-observer variabilities in every study. Because many of you do echo, that is why I thought that is a very important point that you should always consider. Should always do intra and inter-observer variability. So many of you may not be aware how to do it. Like, anybody can anybody say how do you do an intra and inter-observer variability? For example, we are going to study the speckle tracking of children, LV speckle tracking of children. So how do you want to establish intra and inter-observer variability in this for this particular thing? Because it's not a very well standardized parameter in all the echo machines, so you need to standardize it. So how do you do that? No answers? So for example, you can discuss with your statistician and come up with a number that needed to be studied. For example, most of the time for such a study, 10 patients should be good enough. So take these 10 patients. So the first of the one operator, the primary person, that is you, will be recording it 1. The next person, one of your friend, will be recording it 2, and you will be recording it again 3, and analyzing the data again. Any parameter that we talk about. So for example, in a particular child, we take 10 children for answering this question. What is the intra and intra-observer variability of a speckle tracking S velocity? So you take 10 people, 10 children, you record, your friend records, next person records. Let us see, somebody has answered in chat. The COPPA statistics. So this is how you get into the COPPA statistics. Like so whatever the difference that you and your friend, the variations, that is actually put into a COPPA statistics, and that gets inter-observer variability. The one that you recorded once, and then the one you recorded the last, that will be for intra-observer variability. So always, before starting your first patient, please do this, because that's very important. That is one of the major reasons why majority of the echo papers get rejected. Some fantastic phenomenal work, they don't go to a very good journal because of this small thing. That's a very small thing. Like you must have done 100 patients, but first 10 patients, had you done intra and intra-observer variability, things will be totally different. It goes to a different level. So the most important, I told you, like everything is important. Like I told you, like a protocol writing, most important thing is question. Absolutely clear. Then the statement, absolutely clarity has to be there. What are you studying? Why are you studying? When are you studying? Where are you studying? Everything has to be cleared in that statement itself. And the population, everything has to be clearly defined, and the outcomes has to be clearly defined. Based on all this, the patient's case record form has to be created, especially for a prospective study or even for a retrospective study. This is possibly the most important thing. After doing so many research, maybe I would have published around, say, 100 odd original articles till now. I think we learn. It's a process of learning that we keep on learning, and we all keep on learning by doing mistakes. So we should not do that. I think we should go to the next level, because why should you all do the same mistake that I have done over last 10, 15 years? Because somehow we always teach you how to pick up a TAPVC on an echo, but we never teach you how to do a descriptive study of TAPVC. What should be the CRF contain? For example, in that particular VSD PHF study, it's done by a really nice group of people who have published a lot. But children randomized control study, that is also, for us, it was not a good, it was not very commonly done. So once we did the study, I think we realized there are a lot of lacuna. For example, we took patients' weight, but we didn't use the same scale. So patient's weight was taken at different places, as typically happens in our OPD. So that patient weight was a measure that the journal did not accept. So this is the attention to detail that we have to go to. So heart rate, we noted the heart rate. Okay. So what is this heart rate? Whether the patient is sleeping, whether the patient is crying, whether the children is in mother's lap. These are all small infants. Or can you do a small Holter, a 1 hour Holter, to just check the average heart rate for propranolol use? What is the heart rate variability? So this is the amount of detail that you have to get into. And I think possibly the most important giveaway of this talk, I'm telling you one thing. So always prepare your final output paper and just think of it like, or see such similar paper that are getting published when you're doing the protocol, and make sure that all the relevant information you've collected in your CRF. CRF is the case report forms. For example, this is what you will enter for each patient. Always make sure that you enter each and every variable that is very essential for your final presentation, final paper, final thesis. Oftentimes we miss some of these important ones. I think that is very, very important. So patient CRF spend time, repeatedly go to similar articles that are published and look at all the tables. Look at the tables, whether you have captured all the relevant information in your CRF or not. This all has to be done before your protocol is submitted to ethics committee. So I think the most important thesis statement is that oftentimes we don't answer a question, because oftentimes we don't ask a question and we don't answer a question. The best part of major institution is that it goes through a rigorous process where it is scrutinied. You are scrutinied. There's a lot of scrutiny that happens, and then people may challenge it and oppose it. People may say that what is the worth of doing this and so forth. So I think we have to be focused, and you should be able to answer how to do it and why to do it. So if you are able to answer these two questions clearly with a clarity of mind, I think you should go with that statement. So coming to the particular protocol writing, so always not only protocol, I think this is a very simple, because it is daunting task, especially when you write the first paper or when you write a thesis protocol, it looks very daunting. And oftentimes you cut and paste and do this, do that, and see some seniors and then do this. So always make an outline, prepare a draft, revise it, and then edit it. This I think is very simple. So the outline is extremely important. Like, for example, like if a propranolol for heart failure, what will be my outline for an introduction? So the outline for introduction is, so heart failure is so and so common. VSD is a common cause of heart failure in infants, and there are no proven therapies for heart failure. Why should propranolol work in VSD and heart failure? Because beta blockers have been the major medication for heart failure, and there are no very small, 1 or 2 small non-randomized controlled studies in children have said that it is possibly of use. Okay. Now, how this is going to add? Because there is no randomized control study, we want to be the first major randomized control study of doing heart failure. So you just put the outline, then you can change the this is how you organize your ideas. And then language, you can keep on revising it. And so basically everything, as I told you, like don't keep on straying. So get your thing into a focus and prepare an outline and start writing your protocol. So how do you create that outline? I think many of you have written protocols. So I think I think you have to first of all do a good thorough search, brainstorm with your seniors, brainstorm with your colleagues, brainstorm with most importantly with your mentor, because he has more experience. Make sure that he spends quality time with you in this stage. Oftentimes the seniors have this problem that they don't spend too much time at that stage. So, and create subheadings, and then once you create such an outline, and it's very simple to fill it up, maybe 10 pages, it is easy. One of the important tips is begin early. Your protocol submission has to be done within 6 months of joining. Majority of the places have such a rule. Always start within 6 weeks of joining, not just 1 week prior to protocol presentation or protocol submission deadline. So you always have some deadlines and always follow those deadlines and finish it off. So as I told you repeatedly, that be specific as possible. Everything has to be properly defined. So very good, very nice way of putting things together is what is known as a good research question is what is known as a finer criteria. It has to be feasible. That means it is to be answerable with a robust method. It has to be interesting for you, for the audience, and for the scientific community as a whole. It has to be novel, and most importantly, it has to be ethical, and it has to be relevant for not only you, but for the country that we live in. So this is very important that you look at this final criteria, that is feasible, interesting, novel, ethical, and relevant. So now you have finished your thesis. you finish your thesis protocol, and then your thesis is done, because nothing much happens. Like, you can't modify too many things once the methodology is finalized. Now you have written a thesis. So thesis is generally written in a very bulky fashion. Like in India, like we often write a 70-page, 100-page thesis, which I think a lot of irrelevant things are written in that. But then we need to curtail it. And then in brief, I will mention about how do you convert and what are the ways to convert this thesis into a good paper. So IMRD is a standard format most of the journals follow. So that is introduction. So already we have discussed what you write in the introduction, and material, methods, results, and discussion. Another tips that I would give all the junior colleagues is that if your final thesis and the final thesis paper, in the thesis protocol presentation and the starting of your thesis itself, you can finish off introduction, material, methods, and review of literature part. And I would suggest that prepare dummy tables. Prepare dummy tables and keep them ready, and make sure that all the values that you're going to enter in the dummy table, all are being captured, all are in a standardized fashion in the case report form or not. So once you finish up your thesis protocol, everything is fresh in your mind. Start working on your thesis part and finish off introduction, material, methods, review of literature, and results with dummy tables. So I'm telling you, it won't take extra forgotten which reference, what I had, what, and all those things. It's very simple and very easy to follow. So once you start writing, once you think of writing, I think a lot of papers that what particular thing suits you, whether you want to present it in abstract format in the conference, or whether you want to directly go for journal articles, you can think over it. But most importantly, I think in the next few classes, we will be going into the exact details of scientific writing, various how and how not to write, and all those things. But this is a major second important lecture that I have taken. So I think it is often painful that that India is among the top 3 countries where people are caught for medical plagiarism and medical misconduct, medical research misconduct. It is painful. We have so many patients, we have so great doctors, but we are number of articles withdrawn, number of people caught doing misconduct. We are among the top 3 countries in the world, whereas we contribute minuscule number of research articles compared to the rest of the countries who contribute a large numbers. So I think this has to be ingrained from the day you start writing your paper. So no misconduct is possible, or should be, no system should allow that. Like a dictum says, everyone is corrupt as much as the system allows them to be. I think in India, for long, we have been not looking at the scientific writing misconduct as a serious offense, or scientific research misconduct as a serious offense. I think we cannot get away nowadays. So you cannot get away from informed consent, any deviation from usual practice. So the burden should not be imposed on the participants. The risks are posed on the participant without their knowledge, and without clearly telling about the benefits to the participant, patients, and societies. So they are gone beyond. Like this is the BMJ group's official version. So we have not. I've seen some of the theses where even ethical approval is not taken. People are writing to Annals of Pediatric Cardiology, our official journal, without an ethical committee approval. I think this is the standard. Like we cannot take this standard. Like we need to move on. This is the people have moved on from ethics committee, IRB approval to various other things. So there are. I think before we go to the next level, I think there are various other good resource material available. This is the International Committee of Medical Journal Editors, uniform requirements for manuscript submitted to biomedical journal. I think anybody who wants to pursue academic career must read this series of articles. And there's a very good Equator Network Research Center. I think all of you who want to be ambitious, you want to pursue academic medicine should read this. So this is various experimental studies, observational studies. Everything is detailed. How do you approach it? How should you report it? And all those things are there. So I'm setting the stage for my next lecture. How to write, particularly. So this is, I think, will be the starting slide of that lecture. I think what editors and manuscript problem that editors frequently encounter. So most often than not, poorly written, excessive jargon. I think this is, much more common in, from Indian authors. Like we overemphasize things, or we are overrated with what you have found. It is science, like scientific writing. It's not poetry where you have the freedom of exaggerating things. It has to be real. It has to be straight. And second important thing is inadequate or inappropriate presentation. That is why I repeatedly emphasize, always start when you're planning. When you're planning for your thesis protocol, at the same time, you should have a dummy tables with you. Poor description of design. So think of it, everything except for a language part, excessive zeal and self-promotion, rationale confused and contradictory, essential data omitted, ignored, it's boring, important work of others ignored. These are the common problems that editors face when the manuscripts are presented. Coming to the misconduct part, so three major misconducts that are common is fabrication, falsification, and plagiarism. So some things are voluntary and some things are involuntary. Anyway, it is totally absurd in today's time. You cannot get away. You may become a great professor down the line, maybe some few years down the line. But in today's era, you could be penalized for writing one bad paper out of your 200 papers, one bad paper which had a lot of plagiarism or falsification. You will be gone. You'll be done. Your academic career will end now in today's time, today's era. सब कुछ नहीं चलता है, ये सब बिल्कुल नहीं चलता है. Like fabrication, falsification, and plagiarism, absolute no. Now you can actually, fabrication is not on, like you just cannot do it. And falsification, you should absolutely prevent it, and it should not happen. We should not stoop to that level that for one publication we will falsify some data. Plagiarism, especially when you start writing, you are so much influenced, you just take away one paragraph and post it. Like even I've got articles coming to Annals of Pediatric Cardiology where paragraph to paragraphs have been taken. So now the online good resources are available. Even inadvertently you write something, plagiarism is there, you can easily pick it up. Picking it up has become so easy with so much of artificial intelligence and so much of resources available to the editors as well as the journals. So journals invariably check for plagiarism. And even your thesis protocol, some universities are posting it in the website. There are also independent resources, the NGOs and across the world are actually doing and then bringing it to the notice of the universities. So please avoid plagiarism. So you may not write the great sentences, but write it in your own words. That is most important. So some of the good reads that I think before we set the stone for the next 1 or 2 lectures in this series. So there are good things that, as I told you, like equatornetwork.org has a lot of this kind of information, how to write your first research paper and so on. And this article by, it's a very vague journal, Yale Journal of Biological Medicine, but how to write a first research paper, it is, she has put it together very nicely for a novice to start writing. It is extremely good, like even introduction, what are the questions that you should be answering, and all those things are given nicely. And a very important read is Fundamentals of Good Medical Writing by Groves from BMJ. So these are some very good reads. I think if you're interested in reading, you should read them. So I think in this third way, please stay safe. And thanks for your kind attention. I'm happy if you have any questions.